the really warm introduction, Stuart. But yes, no, today I'm here to talk to you all a bit about the work I did during my dissertation that was successfully published in Nature, in collaboration with Stuart and several others. But here today to talk to you about the new dogma of microbial bile acid metabolism. But before we get into the actual work I performed, we do need to discuss what bile acids are. And since we just had lunch, it's incredibly relevant. Bile acids themselves are hydroxylated sterols. They're built from cholesterol. They come in two primary flavors, cholic acid and chenodeoxycholic acid, the key difference being highlighted here in yellow with the lack of a hydroxyl group. These are then conjugated with glycine or taurine in the liver, and it's a two-step reaction where first coenzyme A is ligated at that C24 carbon by the enzyme bile acid CoA ligase, or BAL, followed by aminoacyl transfer of glycine or taurine by the enzyme bile acid amino acid N-acetyltransferase. This then results in our suite of host conjugated bile acids, where they're then stored in the liver. And upon consumption of a meal, they are excreted into the duodenum, where they circulate throughout the entire small intestine, aiding in various facets such as fat and nutrient absorption, cell signaling, and maintaining overall gut homeostasis. They're then reabsorbed at the terminal ileum, circulating back to the liver for reconjugation and recirculation. This is about 95% efficient. And the process itself is known as enterohepatic circulation. However, the remaining 5% will enter the colon and are excreted with feces. But throughout the entire intestinal tract, these compounds are subjected to various forms of microbial metabolism by our gut microbiota. When I talk about our gut microbiota, this is a diverse microbial community present throughout the gastrointestinal tract. It's comprised of around 1,000 species, at least more recent estimates have it at that. And it's estimated that there is just barely more than one bacterial cell per human cell in your body. Yet when you look at genetic diversity, there are around 100 bacterial genes per single human gene, just really emphasizing the potential diversity of metabolic activity within our gut microbiome. And there are, of course, broad implications within health. Our gut microbiome has been implicated in diseases such as IBD, type 1 diabetes, and even a disrupted gut microbiome has been shown to impact C. difficile colonization, where if you've got a dysbiotic gut, you're more likely to be infected by C. diff. But among all of these other traits and important facets of our gut, there have been decades of research into bile acid metabolism on the side of our gut microbiome. And when I talk about microbial bile acid metabolism, there are a number of ways this is performed. The first is known as deconjugation. This is considered a gateway reaction into further bile acid metabolism, where you're just liberating that amino acid residue at the C24 carbon. These bile acids can then be dehydroxylated, where you might have heard of the organism Clostridium scindens or Lactoclostridium scindens as being the sort of poster microbe for this transformation. These hydroxyl groups can also be oxidized and subsequently epimerized by reintroduction of that proton. However, I'm more interested in this still somewhat novel biotransformation by our gut, where non-canonical amino acids, or those outside of glycine or taurine, were found to be ligated to our bile acids. This gave rise to what I'll refer to as MCBAs, or microbially conjugated bile acids, throughout the rest of the talk. These were first described in 2020 by my PI at Michigan State, Rob Quinn, where he and others within the Dorstein lab found that Enterocloster bolletiae, a gut microbe that has been associated with different mental disorders such as autism, was capable of ligating phenylalanine, leucine, and tyrosine to cholic acid, thus broadening our host bile acid pool by 3 different bile acids. However, since then, and even going back to this original publication, MCBAs have been implicated in host health, where they were enriched in infants with a developing gut microbiome, in addition to patients with Crohn's disease. They've also been implicated in colorectal cancer progression, having variable effects on FXR and TGR5, in addition to promoting stem cell proliferation within these colorectal cancer cohorts. However, one would think that these would be enriched in our large intestine. There's a significantly larger microbial community there, yet Jacob Foles from the Phen lab had shown that these instead are enriched within the small intestine. But there are variable concentrations for each individual MCBA throughout the gut. And then finally, from the Terrio lab, they did show that different MCBAs are able to inhibit, if not prevent, C. difficile sporulation, which is, it just adds to their potential use as drugs in the treatment of C. difficile infection. However, at the start of my PhD, the mechanism behind microbial bile acid conjugation was not known, and I was tasked with figuring out how that was performed. So today I'll walk you through the various aspects of this project that was published earlier this year. So the first part I'll talk about is describing MCBA product and producer diversity, followed by zooming in a bit and characterizing mechanisms behind this conjugation reaction, followed by investigating physiological consequences of MCBAs, both in mouse and a human sleeve gastrectomy cohort. So we've talked about mass spec all day, and you've heard various conversations about it, but I do want to just go rather quickly through a very basic outline of how a metabolomics experiment is run. So I'm a fecal microbiologist, tried and true. Some might say I'm a crappy microbiologist. But the overarching workflow that I used was taking a sample, in this case a fecal sample, extracting it primarily with methanol, followed by running that extract on an LCMS-MS system. We use a Thermo Vanquish and Q-Exactive in our case, where we then measured the mass of these compounds, bombarded them with nitrogen, and then acquired this tandem mass spectrum. We were then able to leverage that and use the Global Natural Product Social Molecular Networking database out in the University of California, San Diego, for annotation and molecular networking. Now, when I mention molecular networking, this is looking at how similar these different molecular fingerprints are to each other, where if there are shared peaks, the more shared peaks there are, the greater the similarity will be between those. And we're able to then take these similarity scores, change all of these molecules into nodes, and generate networks based off of certain cutoffs for similarity. Now, to get into the real meat of the project, the first part of this work was uncovering taxonomic diversity of bile acid conjugating gut microbes, where my work focused primarily on the Lactospiraceae, Enterocloster boltiae as a member of this microbial family, in addition to other relevant gut bugs. So I screened a total of 29 different bacterial strains, 16 of which were within the Lactospiraceae. This work, it was a relatively simple experiment of growing an overnight culture, diluting it to a standard OD in either plain media or media containing cholic acid and taurine. Now, we included taurine because it's not a component of typical media formulations, and I wanted to see if microbes could produce taurocolic acid in addition to just glycolic acid. These were then incubated for 24 hours where metabolites were extracted, and we ran this using our standard LCMS-MS workflow. So one of the first things that you look for in any experiment is a sort of sanity check. And so I wanted to make sure that what I was seeing in this data set was recapitulating what was already published. And indeed, I was able to find all of the different MCBAs that were first reported in that 2020 Nature paper. However, expanding this out to include additional conjugates, we can already see a significant increase in the number of microbially conjugated bile acids present in culture. This isn't in vivo where there is already a broader diversity of metabolites available. And then taking this even a step further back, this was some of the first work to show that our gut microbiome and members of that gut microbiome can produce our host conjugated bile acids, calling into question decades worth of research into bile acid changes as a whole, where, all right, is a decrease or increase in a certain primary bile acid due to human effects, due to the gut microbiome? Now the waters are a little bit muddier, thus warranting further research even into previously published data. But even taking this one step further and including all of the different metabolites that were somewhat spectrally similar, we can see that there is a somewhat overwhelming diversity of bile acids and conjugated bile acids that we still don't know a lot about. Now, taking this back to that initial phylogenetic tree and looking at the Lactospiraceae, I found that when mapping MCBA production against this tree, overall production was somewhat inconsistent across taxa. Now, visualizing the actual MCBAs produced, I reduced this analysis to just the amino acid that was ligated. So certain microbes are able to produce secondary bile acids, which could then also be conjugated. I didn't want that to confound our results. I used break-reduced dissimilarity to compare these and hierarchical cluster analysis to group the actual samples. So to get into it a little bit, we've got our very first group. This was our high MCBA abundance and high diversity producing group. And a lot of these were members of the Lacnospiraceae. Our second group was somewhat high diversity. However, they showed an enrichment of glycine and alanine. Our third, there's a little bit lower production with a serine enrichment. The fourth containing a lysine enrichment. And finally, our low producers having a distinct aspartate enrichment for use in conjugation. Zooming in a little bit, now trying to uncover the actual enzymes that are responsible for catalyzing this reaction. It had us thinking, all right, there are many ways to find an enzyme. There are many ways to cut a cake. Do we want to go down the RNA-seq path and just look for differentially expressed genes? Do we want to look at perhaps performing a transposon mutagenesis experiment, or even look at building a cosmid library and then screening for production on its own? But we already talked a little bit about enzymes that interact with bile acids as a whole. The poster child for that gateway reaction being bile salt hydrolase, where it's responsible for liberating that amino acid glycine or taurine, resulting in free bile acids. This enzyme was first described, or at least its activity was first described in 1967. And around a quarter of strains in our gut contain a functional bile salt hydrolase gene. That's important for colonization, and it is readily horizontally gene transferred. And luckily for me, I didn't have to worry about cloning and expressing and purifying this protein too much, because there's a commercially available one that I was able to use as a pilot. So the next stage of this work was just seeing if bile salt hydrolase on its own could perform the reverse reaction, conjugating bile acids instead of deconjugating them. So again, a relatively simple, straightforward experiment, just incubating this purified enzyme with taurocolic acid and an equimolar amino acid mix across various pHs to see if I could further characterize the actual catalytic activity. Took these extracts, ran them on our LCM-SMS using our standard workflow, followed by annotation through GMPS. And so after a short enough time period of only 15 minutes, we're already starting to see production of different MCBAs by the enzyme bile salt hydrolase in vitro. And when you take this out to up to 2 hours, we can see that activity is about complete at that 30-minute time point. However, one thing you might notice is a distinct enrichment of histidine, which may be due to an inability to remove histidine from cholic acid. Again, bile salt hydrolase is known for liberating amino acids, not necessarily conjugating amino acids, which may explain the variability in the amino acid use within each time point and across each pH. But because we ran this across a broad pH range, we were able to look and actually identify a pH optimum, which is just slightly more basic than, or at least slightly more neutral than the deconjugation reaction at a pH of 5.3. We can start to dig into the catalytic mechanisms themselves behind this transformation. So the dogma behind bile acid deconjugation is that a catalytic cysteine present on bile salt hydrolase will form a bile acid enzyme intermediate, where then water can act as a nucleophile, thus liberating a free bile acid. Our idea is that instead of water acting as this nucleophile, the amine group on an amino acid can function instead. And so when taking this into account and just incubating taurocolic acid with an equimolar amino acid mix and glycolic acid as well, in this case glycolic acid, we do still see that production of MCBAs, or that acyl transfer, where the actual glycine residue is being liberated, followed by attack from the amine of the various amino acids incubated. However, we also saw production of MCBAs just when incubating with cholic acid alone, which is somewhat confusing, as the dogma is that this reaction occurs one way, or at least the deconjugation reaction occurs one way. Which, after scratching our head for a bit, there must be some sort of reverse to this as well, where cholic acid can still form that bile acid enzyme intermediate, allowing for conjugation with different amino acids. So characterizing this activity itself and incubating with taurocolic acid here, this demonstrated linear kinetics, which might be frustrating at first, but it reinforces our model in that the amino acid itself is just trying to outcompete water, where as you increase the concentration of different amino acids, it's going to be more readily able to outcompete water. And we saw the exact same thing when we used cholate alone. However, the rates of transfer are about 10-fold lower, further supporting that, yes, cholate can form a bile acid enzyme intermediate. It is just significantly less favorable. Now, we then wanted to see, all right, what other characteristics of the enzyme impact MCBA production? How does the active site in particular impact this? So we already covered that production across microbial taxa is somewhat inconsistent. But when we actually look and map the biosalt hydrolase genes present on these microbes, we can, and then map based on the actual producer group. So again, the lower the number, the better the MCBA producer. We can see that there are 3 distinct clades that form. And then looking even further at the actual amino acid sequence, we were able to identify an asparagine at position 82, where for our high producer BSH group, that cluster 1, that was an asparagine, but for the second best producers in group 2, it was instead a tyrosine. Again, asparagine 82 is associated with high MCBA abundance and high diversity, with tyrosine as low abundance and an increase in small amino acid use. So looking at the actual crystal structure of biosalt hydrolase, we can see that indeed this asparagine residue interacts with taurocolic acid, in this case, within that active site. And so in order to fully tease apart some of the nuances of this, I did end up cloning a biosalt hydrolase gene from Clostridium perfringens into E. coli, followed by performing site-directed mutagenesis to see if I could impact the overall MCBA production in vivo. Now, when looking at the fold change in MCBA abundance between our wild type and mutant, we do see, again, some of those similar trends between group 1 and group 2, where for the mutant itself, there's an enrichment of alanine and glycine conjugation, again, those small amino acids, with a reduction in amino acid use for all other amino acids detected, where it was reduced, if not completely ablated. And now as we start to look at the actual physiological impacts in vivo, one question arises, all right, are these compounds antimicrobial? You've heard a little bit earlier today that some bile acids are more antimicrobial than others. So in order to test that with this suite of MCBAs, again, a fairly simple experiment of growing these bacteria overnight, incubating them in a 1 millimolar solution of various MCBAs or media alone, and then looking at the area under these growth curves and computing the fold change in that area. So across 3 replicates, and these are the average fold changes between our treated and untreated controls. This seems like there's a lot going on right now. However, I want to draw your attention to Enterocloster boltiae, where there is a slight detriment when grown in cholic acid alone. However, it looks like it grows much better, or at least somewhat better, in every other conjugated bile acid provided. But as I'm sure you've already noticed right now, there are 2 distinct microbes that had significantly reduced growth in free cholate, in addition to hydrophobic amino acids used in this conjugation. And it's very clear to see, even just when looking at the growth curves themselves, that growth was nearly ablated, especially for Peptostreptococcus anaerobius, for those hydrophobic conjugates. Now zooming out a lot and looking at the impacts across the entire system, we shifted then to using mouse cohorts, with our first question being, all right, we know that bile acids circulate 4 to 10 times a day as you're eating meals. Do these MCBAs also enter enterohepatic circulation? So for a small pilot experiment, we mixed all 8 of the MCBAs we had on hand in 1 dose, trained mice to eat a peanut butter pellet, and then fed these MCBAs to the mice through that peanut butter pellet. Bile acids themselves are fairly hydrophobic, so this is a much more consistent way to deliver those versus a corn oil gavage or something along those lines. So I do want to give a quick shout out to Max Okros. He was the mastermind behind revamping this peanut butter pellet feeding method. So it was originally developed, again, to deliver hydrophobic compounds, and he was the first to propose to use this for our mice. And so these are the results of that 8 MCBA mixture, where we can see an enrichment within feces of all of these MCBAs within 24 hours. In addition to observing all 8 MCBAs that we gavaged outside of the gastrointestinal tract, suggesting that they are entering enterohepatic circulation and traveling systemically. But as I'm sure you'll notice initially, there was some de novo synthesis in controls, or what we believe is de novo synthesis of these MCBAs. However, most groups didn't know to look for these in their tissues prior to this analysis. So this is likely happening throughout bile acid research itself. However, one of the things that was great is that we were able to detect phenylalanine and serine conjugated cholic acid in all of the tissues that we sampled, which we then followed up on with a much more in-depth experiment with additional controls. Again, training 6-week-old mice to eat peanut butter pellets, feeding them a peanut butter pellet containing an MCBA or an equimolar bile acid amino acid control, in addition to taurocholic acid and an equimolar control to look at how just simply feeding a bile acid would impact overall bile acid concentrations in the mice. And this was carried out for 10 days. So when we look at our phenylalanine or our phenylalanocholic acid fed mice, we do see a distinct enrichment within the gallbladder. This is probably perhaps the most bile dense tissue within our bodies or within a mouse body, in addition to within the duodenum. However, when we looked at the serine conjugated cholic acid results, we can see an enrichment across all of the tissues that we sampled, further supporting the entry of these compounds into enterohepatic circulation, in addition to movement throughout the body. And finally, translating this to something a little bit more relevant and looking at a human cohort, in collaboration with Stuart and the folks at Corewell Health, we had access to a sleeve gastrectomy patient cohort, where this sleeve gastrectomy surgery is a treatment modality for obesity and type 2 diabetes. And it's been previously reported that there is a decrease in total bile acid concentration within feces and other tissues following the surgery, in addition to a remodeled gut microbiome. So there were 45 patients total. We had to exclude one due to a lack of a second sample being collected. And we primarily compared a baseline fecal sample to a 3-month post-op sample. So again, after extracting and running those samples using our standard workflow, a good sanity check is, all right, can we see our human primary bile acids? And we can, and the concentrations are somewhat consistent between groups. Looking beyond that, we can look at some of the more well-studied secondary bile acids, and we see, all right, we can detect those as well. And this is already a fairly diverse suite of bile acids within these samples. But then when we look at all of the different MCBAs we were able to detect, we can see just how diverse our host bile acid pool is. And I hope that with this slide in particular, I can impart upon you the underappreciated diversity of our bile acids. Now, breaking this down into classes and just trying to identify what's different, when we look at primary and secondary bile acids, we do see that there is a decrease, or at least a slight decrease in concentration between the 2 time points. In addition to recapitulating and further supporting the decrease in total bile acids following the sleeve gastrectomy. But one of the things that we uncovered within this is that the most significant change in bile acid classes was for microbially conjugated bile acids. And indeed, this may be what's driving that decrease in total bile acids themselves. So to go through a couple of conclusions of this work, in this, I identified bacterial biosalt hydrolase as an enzyme responsible for conjugating bile acids, and that this MCBA production is performed by very taxonomically diverse bacteria. Again, biosalt hydrolase is rapidly horizontally gene transferred. And then finally, this is some of the very first work to show that these novel bile acid compounds can circulate throughout the entire body. And it is an active area of research as to why they are circulating around, or at least what they're doing throughout the body. So with that, I want to thank not only the sponsors of this event, but I want to thank Stuart for the invite to talk, in addition to everyone at the Quinn Lab. Tony, thank you for your help as well, running the actual mass specs, my mentors, and then both biocrates for supporting this, and Panome Bio for taking me on as a multi-omic analyst. So.